to give a special thanks to Natasha and Jason for setting this up again and reiterate the sentiments of other speakers that we really do wish we could be out in the field with you guys today. Happy to be here to be able to do what we can. And I'm also happy to report that through this entire COVID shutdown, we have kept the Plant Disease Clinic open for business for turfgrass managers and other folks in the state of Florida and beyond to process samples. And so I want to also thank my staff for continuing to do that for us and coming in one at a time to get the job done for turf managers and also the rest of the clientele that we have. We went from about 13 folks in the clinic on any given day to, at the beginning of the shutdown, one person in the lab at a time. So we get about 6 samples per workday of turfgrass in the clinic, and they can stack up pretty quick. So they've done a fantastic job, and I'm really proud to say that they kept it going for us, and it's getting a little better now. So it's going to continue to get better as we move along. All right, so today I'm going to talk a little bit about a couple of disease issues that are going to be coming up in the next few weeks. One of them is a viral disease of St. Augustine grass. I'm going to briefly talk about also large patch disease, which is just around the corner. It's time to apply in the panhandle, and it will be shortly time to apply preventive fungicides in Central Florida in another couple of weeks. So briefly mention that, and then just give a brief update on some personnel changes within the IFAS Rapid Turf Diagnostic service that I manage. So Mosaic disease is the only viral disease of turfgrass that we deal with here in Florida. It's a disease caused by a sugarcane mosaic virus. It's been around for a long time, but within the last few years, last 7 or 8 years, we've had new symptoms develop that we're calling lethal viral necrosis that has caused a larger problem for us, particularly in Floratam lawns that have been showing symptoms that go beyond the mosaic. We'll talk a little bit about what those symptoms are. We'll see a map of where the distribution of this disease is. It's somewhat spotty in the areas that it does occur as far as in the lawns that it affects and the neighborhoods that it affects. We'll talk a little bit about that, and then some management options. And again, we're going to talk about a little bit about trying to prevent spread and some of the variety testing that we're doing to determine which of our varieties we can use to replace Floratam in these lawns and communities that are affected. That disease is going to be starting to show up in Palm Beach County within the next couple of weeks. We'll start getting the phone calls, and so I wanted to briefly go through what it is that we're likely to see, what we know about it, and what we can do in response to lethal viral necrosis on St. Augustine grass. So many of you may have heard of another disease, viral disease of St. Augustine grass called St. Augustine decline. That's caused by Panica mosaic virus. That's a different disease, a different virus than we're going to talk about today. Most of, if not all of the varieties that we grow in Florida, including Floratam, are resistant to Panica mosaic virus. The virus that we're talking about today that causes mosaic and lethal viral necrosis symptoms is sugarcane mosaic virus. The virus does affect sugarcane, but it has also been known to cause a disease on St. Augustine grass for the last 50, 60 years. Within the last 10 years, it's caused severe symptoms on Floratam St. Augustine grass that go beyond the traditional mosaic or broken yellow lines of symptom that has traditionally and historically been reported on the grass. It also affects Bermuda grass, but not severely. Some species of paspalum does not affect Bahia grass or zoysia grass that we know of. So this has been around for a long time. Within the last 10 years, it has developed to be a problem in Pinellas County and South St. Petersburg. It's spread up through Largo area, and in the eastern side of the state has been a problem in Palm Beach County, where it has spread, again, not in every community within these counties, but it's spread from Palm Beach up through Vero Beach, and then down through the Keys and around the tip of Florida to Naples. So it has spread wide and far on the East Coast. It has been, again, pretty spotty in the distribution and where it occurs, but where it does occur in high-end communities that are typically mowed by one or two different companies, it has been very problematic and has caused some major losses for folks. So this is a symptom of mosaic. Mosaic in a grass represents or presents as broken yellow lines within the grass blade. This is kind of difficult to find on varieties that are not Floratam. If we go out into a palmetto or citra blue, this is what we're likely to see, but only if we get on our hands and knees and look through a lawn or landscape that's infected with sugarcane mosaic, we'll find a small percentage of blades showing this symptom. In Floratam, that spreads, causes a more severe mosaic that will affect more of the leaf blades within that lawn. And then whenever we reach the period of the year where we start to receive cool evening temperatures down in the 70s and maybe the 60s for the first time for the year, we'll see these mosaic symptoms progress on the Floratam variety to a necrosis or a brown death of those cells that were previously yellow. And so this is sort of the first golden mosaic transition from the yellow to the bronzed look of lethal viral necrosis. This continues to get worse. Eventually, the affected tissues die systemically. Whatever is connected to this stolon that this shoot that's infected with sugarcane mosaic virus is connected to, those other shoots and roots will die as well. Not 100% of the lawn dies. Here's a lawn that was killed by sugarcane mosaic virus. It was reinstalled with Floratam sod. The second time or after you install the lawn in an affected, or the sod in an affected lawn, the grass tends to die a little more quickly and more severely than in the first development. In the first development on a lawn, it takes 2 to 3 years. The Floratam will slowly thin. Weeds will become a problem. Palmetto, other varieties of St. Augustine grass will be apparent in that they will not die like the Floratam. This is a lawn in Pinellas County where there was a contaminant probably in the sod installed 20 years ago when these lawns were established. The Floratam here is pictured on either side of this section of grass that's not Floratam, the not Floratam grass that did not die. This is what it does to Floratam. You can also see some mower streaks or mower tire tracking lines. That's how one of the ways this virus is spread around is by mowers, and the tires grind in the wet plant sap with the virus particles into new areas. And where we have communities that are mowed by shared equipment, that's one way that this can be efficiently spread. Unfortunately, there's not a lot we can do about that. We'll talk more about management options a little bit later. So again, it's contained to some neighborhoods in Pinellas County. It has spread from the southern tip of Pinellas County further north, as I mentioned, in the county, but not every neighborhood, certainly, and not every lawn within neighborhoods are affected. Those that have Floratam St. Augustine grass as a monoculture are most severely affected, and those that die, of course, are the ones that are brought to our attention. Outbreak in Palm Beach County has spread again across the county, jumping from neighborhood to neighborhood, and then again has spread up and down the east side of the state, reaching Glades, Collier counties in 2019. Hillsborough is a misprint there. That was a sample sent from a Hillsborough client that was collected in Pinellas. My mistake for not catching that. So it is again in Glades and Collier and has just reached there. But the distribution does vary quite a bit from these different states, with Palm Beach County being the most severely affected, Pinellas being second most. We had detected the virus. We know it does occur in the northern part of the state, in Alachua County, Columbia, and Clay, but it doesn't cause the same sort of symptoms of lethal viral necrosis. It hasn't been as severe or as far spread within communities as it has in South Florida. Again, Pinellas and Palm Beach being the two most affected counties. The cause of the lethal viral necrosis symptom is not yet fully understood. The sugarcane mosaic virus is always associated with this problem. A second virus, as I mentioned, was suspected to play a role. We have not been able to confirm that in the lab, my lab, and the virologists that I'm collaborating with labs. And so it is beginning to appear that sugarcane mosaic virus can, in fact, cause this without the second virus, this lethal viral necrosis. From a practical standpoint, it doesn't really matter for us. They're both still going to be spread by mechanical means. They both have insect vectors, but it would be nice to know for us from a research perspective and standpoint so that we can screen for resistance and do some additional research. There does appear to be a temperature trigger for the symptom to change from mosaic to lethal viral necrosis, and that is going to happen in the next couple of weeks to month in Palm Beach counties, and so be on the lookout for that. Pinellas and surrounding counties. If you're in a county not listed and you think that you suspect you may have something like this in one of your lawns, landscapes that you manage, please reach out to us, contact us. We'd like to know where it spread and offer some advice and help there where we can. So only Floratam develops lethal viral necrosis. When we did some research with virologists doing inoculations, we had 3 different Floratam genotypes that we tested, and only 2 of those developed lethal viral necrosis when we inoculated them with sugarcane mosaic virus. We brought this to the attention of Dr. Kevin Kenworthy, and he established a research project, a grad student who's looked at sod product genetics. I don't have a lot of time today to go into it, but the preliminary results from Dr. Kenworthy's research have confirmed what we observed was that some Floratam sod is in fact not the Floratam genetic variety in its entire makeup, and other sod products that are sold under different names may contain some amount of the Floratam genotype within them. So Floratam is an aggressive variety, and it's difficult to distinguish in mixed genetic products. So certainly where a sod is installed that has Floratam, it will, and lawn has died from lethal viral necrosis and sugarcane mosaic virus, it will die again. Again, it's affected by this problem. And so we recommend not using Floratam to replant, but it is difficult to determine for sure if what you're purchasing, or for the case of turf producers, what you're selling is in fact free of the Floratam genotype. So, so what do we do about it? Quickly, don't regrass with Floratam if you have this problem. Select another variety of St. Augustine grass. Palmetto has been one variety that Dr. Kimmer's research has shown is a good, consistent variety within the genetics of the sod products that we purchase that are labeled as Palmetto. Excuse me. Bitter blue is not. Bitter blue tends to, in several cases, contain at least some part Floratam, and so I think we have stopped recommending bitter blue where it cannot be assured that the bitter blue does not contain Floratam. So Floratam has large purple stolons. Still, visual ID is tough to do. So Soja grass and Bahia grass are not hosts again, but and they offer an option that will not die from sugarcane mosaic virus, so won't get it at all, and they also won't serve as a reservoir for the virus. Okay, so regrassing options there include sodding, sprigging, letting the grass die, and letting it transition to a new genotype by plugging. All of those are options. Very quickly, I want to give you an update on the Plant Diagnostic Center here in Gainesville. We offer diagnostic services and have for many years, as I mentioned. The Rapid Turf Diagnostic Service is the service that we have dedicated to turf grass and a rapid turnaround. Our full-time diagnostician through the middle of this year was Mr. Max Duncan. He was also my student working on a research program. 2019, we did over 900 samples. We use specific media to have a rapid turnaround and give research-driven management options with those diagnoses, and it costs a little more than a standard service. The 2020 update is that Max has moved on to an industry job where he's making the big bucks, and we're really happy for him and definitely thank him for his service to the program. We have a new student, Rapid Turf Diagnostician, Mr. Nick Goltz. If you've sent in samples recently, you may have heard from Nick, and we're really happy to have him on board. What do we get? We get a lot of Bermuda grass, but St. Augustine grass is creeping up as well. Most of these Bermuda grass samples are golf course turf. We do receive a lot of high-end St. Augustine samples in addition. Jason and Natasha, how am I doing for time?
You have a couple minutes.
Couple minutes. Okay, good. Thank you. What do we see in the sample? Real briefly, take all root rot and Bermuda grass decline are root pathogen issues, diseases caused by Mannomyces graminis and other relatives. Those are usually at the top of the list with Pythium, and that was no different this past year in 2019. We saw a lot of root disease problems, a lot of Mannomyces, Pythium, gray leaf spot came in on those St. Augustine grass samples, as well as curvularia and bipolaris diseases of the Bermuda grass. Leaf and sheath spot is caused by Rhizoctonia zea or Whitea cercinata variety zea. That is a problematic disease for our golf course superintendents, and then the list goes on with and including sugarcane mosaic virus continuing to be an issue for us that we're testing, identifying, and then making management recommendations for in those counties affected. We get a lot of different things that come through the clinic, and I wanted to stress here at this point that it is important to get a good diagnosis, whether it's my lab or another lab, of a disease problem or a problem to make sure it is disease before we take management actions, because that's the only way to really have an efficient and effective management strategy. Take a sample, just some general tips here on how to get the most out of a diagnostic service. Take a sample as soon as you notice any kind of symptoms. If something's wrong, the sooner you get a sample to a lab, the better. And before fungicide or any other pesticide is applied, is better for us and it doesn't slow us down in the lab. A cup cutter size or a coffee can plug type size of grass is generally what we need. Smaller amounts of tissue can be difficult for us to work with. So about 4-inch plug works the best with some soil, minimal amounts, roots, stolons, and leaves, a complete turf plant. Because even though the problem may be on the top, the issue and the pathogen may be chewing on the plant down below. So take the sample from an edge of a disease symptom, shoot for 2/3 of that sample to include disease tissue that's showing the sample symptom that you're concerned about, and 1/3 healthy border tissue, so that we'll be sure and get a good snapshot of the pathogen as well as any saprophytes that may be following it up. Send the sample expedited or drop it off if you can. We have a drop box outside the clinic that's still functioning. We are currently closed to unscheduled visits, however, so you can drop it off in the cooler outside the door if you do come to campus. We are here, but we have the doors closed for now. Send an email and attach pics of the field signature. What does that disease look like to you as you walk up to the turf? That can help us narrow down what it might be. With that, I want to turn it back over to Jason. And again, thank you, Jason and Natasha, for organizing this and for your patience and help.